we have known about megaspore mother cells, and now I will describe about placenta. After this section, I think you will be able to understand the whole things. Female gametophyte. Female gametophyte is known as embryo sac. Okay, the functional megaspore when developed into embryo sac. This process, that's mean embryo sac formed from megaspore. This process is known as monosporic development. It is very important. Okay, monosporic development or process of formation embryo sac from a megaspore. মেগাস্পোর থেকে এমব্রায়ো স্যাক প্রডিউসড হচ্ছে। In the meiotic division from a megaspore, 8 nucleate stage race, and then 3 nucleate move this place, this sides, and other 3 in that place, and 2 from both of them 2 nuclei present in the midway. This is known as polar nuclei. Okay, this is known as polar nuclei, and here 8 nucleate stage we can see, but 6 of 8 nucleate are surrounded by cell wall. Okay, here 8 nucleate present, but these 3 are covered by cell wall, and these 3 are covered by cell wall, but this one and this one have no cell wall. They stay in the middle part of embryo, and it is fused next time and known as polar nuclei. After fuse 2 nucleate and made a polar nuclei, and then microfilar end part, these 3 cell we can find, and I have told that opposite of microfilar end, here I have seen this is microfilar end, this is classal part. Microfilar end, this 3 cell present. Okay, in this 3 cell, 2 cell are synergids, and 1 cell is egg, which is big, more than 2 in size. This egg is more bigger than 2 synergid cells, and you have to always remember opposite of microfilar end part, classal end. I have seen classal end. In this place 3 cell are present. This 3 cell is known as antipodal cells. Okay, here if you have to find out antipodal and egg, if you are called to find antipodal part and egg, then first of all you have to find out microphylar end and then classal part. If you can denote this part, then you will be able to find out synergid cells, egg and antipodal part very easily. So always remember microphylar end present synergid cells and egg, and classal part antipodal cell present. Okay, it is very simple. Here take it very simple, then it will be simple to you. First of all, we have seen 8 nucleated stages here, 8 nucleated stages, but in this case we have find out that 8 not 8, 7 nucleated stages, because 2 are fused here. Now we have finished all part of male and a female plant, especially flowering plant. Okay, now we have to go next. Pollination is a breeding process. Autogamy. In some same flower, when pollination happened in same flower. Okay, this is known as autogamy. Syncropy of the pollen grain release and stigma. Anther and pollen is in very near. Example viola, common pansy, oxalis and commonelina. Chesmogamous, one flower to other flower of same plant. Here realize same plant but flower are different, then it is known as chesmogamous pollination. And when this flower is not free, it will when flower are not open at all, this part is known as clistogamous flower. when pollen transfer, same type of plants but different flower, same type plant but different flower, this is called geitonogamy. Transfer of pollen from anther, transfer of pollen from anther to stigma of a different plant. So when pollen go different type of plant flower is known as geitonogamy. This is two types of flower. When this flower pollen go here, it is known as geitonogamy. Agent of pollination, you have to study yourself. It has some characteristics, you have to remember it and you have to study properly. Okay.